And I think this is a really important area because I'm aware by talking to parents and young people in my clinic who are in a similar position, not currently having access to modulators because they don't have the right mutations, that their kind of feeling is that there might be just one type of mutation that isn't responsive to modulators, and therefore one new type of genetic therapy will definitely work for them. And that's not necessarily the case, but it does depend a little bit on exactly what type of genetic therapy we're talking about. So, for example, some people who can't currently access modulators have stop mutations, which is where no protein's produced at all because there's a premature signal in the machinery that stops the protein being made. Now they would benefit from gene therapy. They benefit from mRNA therapy because those therapies aren't mutation specific, but they could also benefit from more specific therapies. For example, the therapies called read-through agents that can read through stop codes in the message. So that's one group of patients that are slightly different and could have potentially more options for these new genetic type therapies. There's another group of people who currently don't have access to modulators that have what we call splicing mutations, and that's where when the message is being processed, it's cut up in the wrong way, and then we don't get a proper code to produce the protein. Now, people in that category could potentially be treated by very short sequences of RNA that could be inhaled. And clinical trials are already started late end of last year for people with that kind of issue. Now, the difficulty for people in that category is there's lots and lots of different splicing mutations, and you need a very specific short sequence of RNA, which is called an antisense oligonucleotide, or ASO. And so they're going to have lots of bespoke potential models to try and treat people in that situation. The advantage of very short sequences is that they potentially could be absorbed into the lung more easily. And my understanding of the current clinical trials of those agents is that the scientists working on those think that they'll be absorbed actually without coating them either in fat globules or using a virus or other sort of vector. Then we get on to the what we call the agnostic therapies that could work for any mutations, such as longer sequences of mRNA that could code for the whole CF protein, or indeed the gene therapy that could code for the whole protein. And they could work for anybody, regardless of mutation, which is good news. The worst news about those sorts of therapies is that delivery may be harder to the lung, and they need wrapping in some kind of delivery system, such as fat globules or using viruses to deliver them. And we're getting more and more promising results, certainly in the laboratory, that we've got systems that are going to overcome the barriers that are in the lung. But if we can overcome those barriers, then the more gene agnostic types, so the mRNA therapies or the gene therapies, look like they're going to be because they can work for anybody regardless of mutation. Those are the most promising agents coming through. So I hope that's a brief synopsis of the different types.
In terms of the DNA therapy and the mRNA therapy, we're always thinking of its replacement. So if your car breaks down on the motorway, this is saying someone comes along and gives you a new car, but the old car remains on the motorway. The difference here with the gene editing is that what they're trying to do is to do the sort of RAC and come along and actually repair the old car and sort that out for you. You don't need a new car. So I think that's the sort of differences. And maybe we should also distinguish clearly between the DNA and the RNA, because the DNA has got to get from the outside through the cytoplasm and into the nucleus. So there are several barriers there. The RNA only has to get into the cytoplasm because that's where it works. So you're saving one important barrier. So why on earth would anybody not just do RNA? And the answer, I think, has been, first of all, you will understand that the RNA is broken down quite quickly because that's the way the cell works. And so you're going to have to keep on giving it. And it comes back, Lizzie, to your point about cost of goods. And certainly until recently, RNA has been not that stable. It's definitely getting better, but it hasn't been that stable. So you're talking about delivery every day or every 2 days or every week, that sort of thing. With the stuff that goes to the nucleus, what you're thinking about is it will last a lot longer if you can get it to the nucleus. And then it comes in two flavors, whether it just floats around the nucleus, so-called non-integrating, which is what the fat globules do, or integrating, which is, for example, what our lentivirus does, where it actually clips into your own genes. Now, that brings with it stability. It gives you very, very long-lasting expression. Just brings with it the slight issue of if it goes into your genes, where does it go into your genes? And if it does it randomly, could it do some mischief? And so, there are balances here to do. But I think that's the key difference between the DNA, lots of barriers, possibly long lasting, and the mRNA, less barriers. You might have to give it a lot.